Good morning, scholars, and happy Thursday. Mr. Schull here. My iPad is charged and ready to go, so I'm going to be making part 3 of our subunit vaccine video series. Really quickly, let's review what we have gone through so far. Our big goal is to create a subunit vaccine. The way we are going to achieve that goal is we're going to get bacteria to make pathogen proteins for us. Because remember, all a subunit vaccine is pathogen proteins. So the way that we collect these pathogen proteins is we use bacteria to do that work for us. So remember the very first video I did where I kind of gave the analogy of my robot grandma. If I wanted my grandma to bake me a pie, I know that she wakes up and she makes all the recipes in her cookbook every day, so I can sneak in the pie making recipe and she will make me a pie. We do the same thing with bacteria. Bacteria are kind of like a robot chef that as long as you give them the recipe, they will make it for you. So we're trying to sneak in the recipe for a specific pathogen protein. That way they make the pathogen protein for us. And that is now our subunit vaccine, which can be given to patients. So our mini goal, what we need to do to get the bacteria to make them for us, is we need to insert pathogen DNA into a bacteria cell. The reason we want to do that is that when the bacteria cell does protein synthesis, it's going to read that pathogen DNA and it's going to end up making pathogen proteins for us. Now, the best way to try to give pathogen DNA to a bacteria cell is by using a plasmid, because we know plasmids are really easily absorbed by bacteria cells. So what we did is we cut the plasmid one time. That opened up the plasmid. We then took a section of pathogen DNA and we cut a gene out of it. This would have been a gene for pathogen proteins. We snuck that gene into the plasmid DNA. The way that we made those cuts is by using restriction enzymes. And restriction enzymes cut at specific sequences of DNA and they create sticky ends. So all 3 cuts, 1, 2, 3, were all made by the same restriction enzyme, meaning that their sticky ends should match up with each other as shown here. We had the sticky ends exposed on the plasmid DNA, and we also had the sticky ends exposed on our pathogen protein DNA. Because they were cut by the same restriction enzyme, those sticky ends lined up with each other, and the pathogen protein DNA was able to be inserted into the plasmid. This is kind of like what you might call the Trojan horse technique. Okay, so the way that they use the Trojan horse to get into the city of Troy, is that what it would have been? I need to rewatch the movie and relearn my history. But what they did was they presented the city with a Trojan horse as a gift, right? So the city was like, ooh, yay. And they accepted the Trojan horse into the walls of the city. And then at night, it turns out there was a bunch of soldiers hidden within that Trojan horse that then were able to get into the city. We're kind of doing a similar thing here. We're going to present these bacteria with this plasmid. And they're like, ooh, I love absorbing plasmids. But little do they know that embedded into this plasmid is a gene for pathogen proteins to be made. So let's move forward from here. I'm actually going to use more space, so I'm going to slide on to the next page. What I'm going to do, though, is I'm going to cut out this drawing, and I'm going to copy it. Oops, no I'm not. I'm going to paste, there we go. And I'm going to bring it, ay caramba. Okay, give me a second here. Okay, let's go ahead. I should probably just pause the video while I'm doing this, but it's too late. Guys, I am slowly learning Notability, but it is taking me some time, I'll tell you that much. Okay, let's duplicate and take this down here. Yay. And make it bigger. Double yay. Okay. Oy. Perfect. Okay. Now that we are ready, let's talk about this plasmid that we've made. Now we call this plasmid specifically a recombinant plasmid. And let's break down that word recombinant. Recombin kind of looks like the word recombine, right? So if you think about it, this is kind of like a recombined plasmid. We disassembled it, we broke it open, we snuck pathogen DNA into it, and we recombined it together in order to make this plasmid. Therefore, it's called a recombinant plasmid. What I want to do really quick is kind of throw things back to this first drawing where we drew out the anatomy of the plasmid, and I kind of want to highlight some key regions or some key genes on this plasmid. The first one is that this plasmid will still have an origin of replication. So let me go ahead and highlight for you here. There's still going to be a region on this plasmid, which is the origin of replication. Now, also on this plasmid, there still should be a gene for antibiotic resistance. That might not mean much to you right now, but it should in a minute, okay? So this plasmid also has a gene on it for antibiotic resistance. So I'm going to say gene for AB resistance. Okay, and now because it is a recombinant plasmid, of course it also now has the gene for pathogen proteins. Yay, awesome, okay. So what we do now is all this plasmid is, it's not a living thing. Remember, it's a circular piece of DNA and its instructions to be able to make proteins. But we need something to make those proteins for us. That something is going to be a bacteria cell that will make proteins. So what we do is, let me draw a little bacteria cell for you all. Dun, 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 dun. Bada bing. Bada boom. Let's give him a little face. There's our happy little bacteria cell, okay? What we are going to do with the bacteria cell is we are going to try to get it to absorb this plasmid. The way that we get it to absorb the plasmid is through a process called heat shocking. So let me go ahead and make this nice and tiny. Okay. Oopsie. So we're going to use a technique called, oops, let me make the pen size smaller. We're going to use a technique called heat shocking. What we do is we really rapidly heat up and then cool down this bacteria cell. What that does is it takes its cell wall and cell membrane and it kind of makes it loosey-goosey for a second. Heating it up kind of makes it so the membrane becomes a little bit more fluid, allowing for things to pass through. So what we do is we heat up the bacteria cells really quick, and what happens while we heat them up is this plasmid should be able to enter into the bacteria cell. So let me go ahead and really quickly make another copy and show that this plasmid is now inside of the bacteria cell. Yay! Why does this matter? Well, let's think about this bacteria cell going through protein synthesis. What is it going to do? The first and most obvious answer to that question is that when this cell goes through protein synthesis and reads the DNA in this plasmid, it is going to be reading the instructions to make pathogen proteins. Okay, so as it reads this gene right here on the plasmid, this bacteria cell is going to start producing pathogen proteins, which I'm just going to draw as little orange stars. Okay, it's going to start pumping out those pathogen proteins. And keep in mind, those pathogen proteins are the subunit vaccine. So guess what? Now we have a subunit vaccine. We can bottle this up, purify it, and now inject it into a patient. Okay. What is the significance of the other two regions on this plasmid, the origin of replication and the gene for antibiotic resistance? So I have one bacteria cell that has the genes to make pathogen proteins for us, but that's just one bacteria cell. That is not going to make a lot of pathogen proteins. I am running a vaccination development clinic. I want to make as many vaccines as possible. I'm going to need a whole lot more bacteria cells making these vaccines. So what I need is for this bacteria cell to grow and divide, right? So eventually, this one bacteria cell is going to turn into, duplicate, two bacteria cells, right? So now I have a second bacteria cell that is able to produce pathogen proteins, okay? The only reason this second bacteria cell is going to be able to produce pathogen proteins is because it has the gene for pathogen proteins. The only way it'll have the gene for pathogen proteins is if it has its own copy of this plasmid. Okay, are you starting to see where this is going? The copy of this plasmid can only be made if the origin of replication is intact. So because this plasmid still has an intact origin of replication, this bacteria cell will also have the copy of the plasmid. Now, there is a chance that some bacteria cells might duplicate, but for whatever reason, oopsie, that's not what I wanted to do. Let's see, I don't know if that's going to be possible. Let's go back and back. There is a chance that some of these bacteria cells might duplicate. I'm going to draw this quickly because I'm also running out of time on my screen recording. Might duplicate, but not receive the plasmid. Okay? If it can't receive the plasmid, it's not going to be producing pathogen proteins for us. So it's kind of useless to us, right? So as scientists, do you think we want to spend the resources keeping this bacteria cell that can't even make pathogen proteins alive? No. So what do? Well, we take advantage of the fact that we know that any bacteria cells that can produce pathogen proteins also has a gene for antibiotic resistance, because remember that resistance gene is on the plasmid. So knowing that, we can actually take these bacteria cells and put them on a large agar plate, like a petri dish. Dun, dun, dun. Right? So this petri dish is going to contain liquid broth, right? LB or agar, and it's also going to contain antibiotic, because the bacteria cells that do have the plasmid will survive on this plate because they have a gene for antibiotic resistance, okay? And so therefore, they're going to be making pathogen proteins, so they'll survive. They're going to be all good. The bacteria cells that didn't get the plasmid and aren't producing pathogen proteins, they don't have antibiotic resistance, right? Because they never got the plasmid that has that antibiotic resistance gene on it. So this bacteria cell is not going to survive on this plate. It is going to die on the plate. So we can actually just erase it. That is how scientists verify that the bacteria have absorbed the pathogen protein gene. If a bacteria absorbed the pathogen protein gene, then it will be able to survive on the plate with the antibiotic because it's paired with that on the plasmid. This should all help you answer this question up here, which is, why is it important that genetic engineers do not disrupt the origin of replication? And I would also add, why is it important that they do not disrupt the antibiotic resistance gene? I'm running out of time on the recording. Have a great day.